Right. Okay, so let's do a summary chart first for this chapter, cell membrane transport, to get an overview of the content of this particular chapter first. Okay, so biomolecules got lipids, proteins, and carbohydrate. Under lipids got phospholipid and cholesterol. Hello, cholesterol. Okay, so phospholipid is the main molecule that form the membrane, because the nature of the phospholipid has got a phosphate head, which is hydrophilic, and fatty acid tail, which is hydrophobic. So therefore, when we put them into water, they will arrange into phospholipid bilayer. Okay, so where the phosphate head will be facing the water on top, as well as the water at the bottom there, and then the tail will be facing each other, so that they'll be facing away from the water. So therefore, we got a phospholipid bilayer. Okay, so phospholipid bilayer. Then when you learn about phospholipid bilayer, we say that the membrane has got this property called the fluid mosaic model, right? The fluid mosaic model. So why is the membrane called fluid mosaic model? Sufyan.
Fluid because lipids are able to move freely.
Very good. The phospholipid can move freely laterally, right? Okay, so very good. So because this phospholipid can move freely laterally, this way, sideways. Okay, why sideways and not up and down? Is because this fatty acid tail doesn't like water, right? It's hydrophobic, so it doesn't want to mix with the water. So it can only move sideways so that it's protected away from the water. So move sideways is called fluid. Just like any water molecules move, it becomes fluid. Now, why is it called mosaic then? Thank you, Sufyan. I've given you 2 marks. Who would like to try? Let me call Sarah. Sarah?
Because there are proteins scattered on their bilayer.
Phospholipid bilayer. Phospholipid bilayer. Very good. So keyword is protein scattered. So it's fluid mosaic model. Very good. Okay. So this is phospholipid. Cholesterol. Cholesterol, you already learned the structure of cholesterol, right? The structure and function. So cholesterol overall is hydrophobic, but it has got an OH group that's hydrophilic. So therefore, when cholesterol sits into the membrane phospholipid bilayer, it will actually stretch from the phosphate head to the fatty acid tail region, where this region here is totally hydrophilic, which is the OH part, and the rest of them are hydrophobic. So it's aligning with the level of the fatty acid tail, right? And it's smaller than the phospholipid bilayer, sorry, smaller than this molecule, phospholipid molecule. So that's why it is slightly smaller. Okay, so cholesterol play a very important role, just like the phospholipid molecule. The fatty acid part, so we are focusing on the detail now, the fatty acid part. This fatty acid part, why am I using red color? Okay, this fatty acid part, right? You learn that it's got 2 more properties for this fatty acid, which is actually, it can be saturated or unsaturated, right? Saturated versus unsaturated. So when it's saturated, you know that the tail will be a straight chain, right? Straight chain. Whereas it's unsaturated, suddenly there'll be a king and it will start to bend somewhere else like that. So this is a king, K-I-N-K. Okay. So this kind of, whether saturated or unsaturated, will actually affect the neighboring phospholipid molecules, whether you can align side by side or cannot align side by side, right? So you've got a king, then they cannot align side by side. They will be aligned like further away because, hang on, I know. So you got this one here, quite far away. Hello, where are you? Here. This one here. Oh, I'm drawing, but it's white color. I cannot see. Give me red color. Right? Okay. So straight chain, bend chain. So this bend chain will push further away. I think I will draw nearer, then you can see. Okay. Straight chain, bend chain. So that's why the spacing here becomes bigger, because of the bending. So the bending doesn't allow the neighboring phospholipid molecules to arrange near to each other. So when they cannot arrange near to each other, that means the neighboring fatty acid chain cannot form hydrophobic interactions. So when they cannot form hydrophobic interactions, they're more free to move laterally. So when they're more free to move laterally, that means they are more liquid. We call it less. So less viscous. Less viscous means they're more liquid. Okay, more liquid. So that is affecting whether it's more viscous or less viscous. Okay. Now, same for cholesterol. Cholesterol itself, right? It's got a structure where I roughly draw only because I cannot draw the chain here. So it's structured something like this. So whenever you've got cholesterol between neighboring phospholipid molecule, like this, it will actually prevent the neighboring phospholipid molecule to be aligned very closely to each other, so that you see this part here cannot form a hydrophobic interaction. This part here cannot form hydrophobic interaction. So it's actually reducing the hydrophobic interaction with the neighboring phospholipid molecules. So therefore, this will actually, cholesterol's main function is to maintain membrane fluidity. That means it's not too fluid, nor is too viscous. So membrane is still fluid, but not too fluid, not too viscous. Why? Why not too fluid? Because it can still form hydrophobic interaction here and hydrophobic interaction here. Okay, I have to draw nearer. It can still form hydrophobic interaction here. Okay, but not here. So therefore, it can still be not too fluid, right? Because it can form hydrophobic interaction, but this part here cannot form hydrophobic interaction. So not too fluid, not too viscous. So why is this property important? Because we don't want the membrane to become so viscous that it will freeze at low temperature, because when it freezes at low temperature, it can break easily. So when it breaks, then the cell will die. Okay? Right. So that is about the structure and property of the lipids. All right. So after that, we also look at the proteins as well as carbohydrates. So when you look at protein, we actually look at the different kinds of proteins that you can find inside the membrane. So can you name me all the proteins I've learned in the membrane? Amar?
Sorry?
Name me all the different kind of proteins you can find in the membrane. Function. Faster.
I don't know.
Don't know. Got so many protein. At least give me one. You learn so many protein membrane already. Faster. Can. Sure can one. Cannot. Okay, open to the floor. Yes, Lo Cesian said channel and give me hang on. Channel protein. What else? Carrier protein. Correct? All these are needed to transport the particles across the membrane, right? You got the glycoprotein, right? Protein with carbohydrate chain attached to it. Very good. Then you've got the pore protein. Pore protein is actually channel protein, pore protein, right? Because it creates a pore across the membrane. It's actually channel protein. Okay. What else? Receptor. Okay. Of course, you got another one. It's got extrinsic protein, means protein that is outside the membrane but attached to the membrane, or intrinsic protein, right? Okay. And then you've got the what? Partially transmembrane protein and transmembrane protein, right? All these are these are the terms for the location of the protein. Extrinsic, intrinsic, transmembrane, partially trans are location. Whereas the function is channel protein, carrier protein, glycoprotein. Okay. Of course, some of you will say it's antigen. Glycoprotein can function as antigen. Okay. But the nature of the molecule is glycoprotein, function antigen. Okay. Right. So of course, there are some more. I've got enzymes enzymes inside the membrane, electron carriers, right? Electron carriers, Okay, so these are the proteins. Now, carbohydrate, there are only two carbohydrates they have learned. So either the carbohydrate attached to the protein, so they form the chain, and this carbohydrate chain is always facing outside the cell one, right? Let's say this is a membrane, right? Okay, with phospholipid bilayer. This carbohydrate chain can only be found outside the cell, not inside the cell. Okay, and then if you say, okay, I've got glycoprotein because I got glycocarbohydrate chain attached to the glycolipid. So this glycolipid, this glycoprotein. Glycolipid means you got glyco means carbohydrate chain attached to the lipid, which is phospholipid. So glyco is this part here, the carbohydrate chain, and lipid, the phospholipid. Okay, glycoprotein means carbohydrate chain attached to the protein. So both can function as the antigen, isn't it? Both functions as antigen. Okay, what are the two more functions of this one? Can you tell me, please? Gan Yuhan. Two more functions of these two molecules. I give you 2 marks for your response just now.
To receive chemical signals. Is it?
Yes, very good. Receptor, to receive chemical signal. Very good. I like the term chemical signal is 1 mark. Okay, very good. Receptor. One more. I give you mark. Okay, one more. I give chance to other people. Type your answer. One more to go. One more. Hello, one more. Self-recognition and self-recognition. Very good. Lo Xi Xian. Today is very basic, but go through a lot of things. Okay. So this is for what? Lo Xi Xian say what? Lo Xi Xian said self-recognition. Self-recognition is actually antigen. Antigen allow for self-recognition. Okay. Self-recognition using antigen. Okay. And cell recognition. Cell recognition is also using antigen. Okay, because you know that, okay, this is my own cell. Okay, this is my own cell, means cell of the body. How do you know that this is a cell body and this is not, let's say, for example, this is not Mycobacterium tuberculosis, because you use the antigen. Okay, cell recognition. Cell recognition is cell recognition. Cell recognition is when the cell is growing to form tissue. Right? How do they know they're supposed to be there and they cannot grow beyond that layer? Is by cell recognition. Cell of the same type, for example, epithelial cell will be located here. Okay, and then you've got connective tissue here, say for example, and then you've got a fat cells here, for example. So they remain in that layer because of the cell recognition. When they grow, they know that they're supposed to remain here. Right? When they touch this cell, they know that this is a different kind of cell. They will not grow to this layer. By cell recognition. Cell of the same type to form the same tissue will grow in the same area because of cell recognition. They know where. Okay. So basically, antigen and receptor. These are the two basic ones. And then antigen will help in cell recognition. Okay. Right. Oh, why? Finish it here. Okay. Next. So this basic biomolecules, right? Yes.
Can you let Chloe in? She's outside.
How long she's outside already?
I don't know.
Okay, right. So thank you very much. So this is the first part, all about biomolecules. First part. Second part of this chapter, we learn about the transport across membrane. Okay. Transport across membrane. Okay, so therefore, under transport across membrane, there are a few mechanisms that you learn, right? The first one is called the simple diffusion. Then you've got the facilitated diffusion, right? Under facilitated, after that, you've got the active transport, right? Then you've got the number 4, the something not correct already, osmosis first. Osmosis, then followed by what did I say just now? Active transport, correct? And then number 5 is bulk transport, right? All the transport. This is the second section of this topic. So this second section. So if I'm to do the summary chart, then I'll draw the line, okay, to, give me red color line. So I'll draw the line, okay, to join everything that I've learned together and divide this topic into 2 sections like this and become very clear cut. And under biomolecules, I will have, right, I'll draw the line, right, to show that there are 3 biomolecules. These are all you know how to do, right? Okay, so that's why summary chart is very helpful. When you do revision, you do like this, your understanding becomes very crystal clear, right? Do it this way. So simple diffusion is when the molecule passes directly across the membrane, right? Because the molecules are very, what happened? Hello? Oh, hard saying, oh, I thought gone case already. Okay, here. Is this bigger already? How come it doesn't go bigger? Okay, very good. Yeah, very good now. I'm recording actually. Okay, right. Okay, where am I? Okay, so when you want to talk about molecules that can pass through membrane, all right, then we have to start to use the term membrane is partially and selectively permeable. So this is when you start to learn the concept of this term. Yeah, right. Membrane is partially as well as selectively permeable. Okay, so partially permeable means among all the molecules that you have, some of them can pass through, some of them cannot pass through, right? So what governs the ability of molecules to pass through is to do with the property of molecules, right? So the molecules, whether it is size, big or small, whether it is charged or uncharged, okay, charged or uncharged. So basically, these are the 2 main things that govern the ability of molecules to or ions to pass through the membrane. So when it's too big, cannot pass through. When it's charged, cannot pass through because this region of the membrane is hydrophobic. So anything that's charged cannot pass through. Anything that's uncharged and small will be able to pass through. Whether it's hydrophobic, hydrophilic, it can pass through because it's not charged and it's very small. For example, the water molecules, the oxygen molecules, the CO2 can pass through directly like this. Okay? So those molecules will pass through by simple diffusion. Now, of course, the direction it will move, whether it's going down, going up, it depends on the concentration gradient. So what governs the movement of molecules across the membrane is actually the property of the molecule plus its concentration gradient. Concentration gradient. Okay, so that governs the direction of movement. Then you also learn about faceted diffusion, whereby when the size is too big or it is charged, the molecule cannot pass through the spacing between the phospholipid molecules. So it needs something to facilitate it to pass through. So either channel protein for ions to pass through, like sodium ions, okay, or it needs a carrier protein. Carrier protein is much bigger, right? Much bigger. So let me remove this thing. So carrier protein will be something like this with now we cannot call this active site, this binding site. Active site only for enzyme. Okay, for molecule with complementary shape with it to actually transport across. So again, the direction of movement depends on concentration gradient. Which part is higher than the other? So faceted diffusion. So faceted diffusion will either use carrier protein, because this is the first one you learn, okay, or channel protein. Right? So either or. Then, of course, we move on to learn osmosis. Osmosis is all talking about the movement of water molecule across the membrane. Now, when the water molecule is moving freely, not across the membrane, but inside the beaker, that movement is called, it's not called simple, it's called diffusion. Simple diffusion is movement directly across membrane, like this is called simple diffusion. But without the membrane, it's called diffusion. Okay? Like the water molecules are moving freely because of kinetic energy. So this is called diffusion. Okay, but the moment you put a partially permeable membrane and allow it to pass through, when it passes through like this, it's called osmosis. Okay, passing through membrane, you must use the term osmosis already. But if it's not passing through the membrane, but it's moving freely like this, it's called diffusion. Okay, for water. So this is where you have to pay attention to the detail to know when to use which one. Very crystal clearly, you need to know. Active transport means against concentration gradient, right? So let's say, for example, diffusion is going downhill, right? This is higher concentration, this is lower concentration, okay? So going downhill is called diffusion. Going uphill is called active transport. So because of going uphill, the cell will need to use ATP. So active transport is with the use of ATP. Okay, example, they learn sodium potassium pump. Sodium potassium pump, right? It's a carrier protein which has got binding site for sodium, potassium, as well as ATP, because three of them must be able to bind to this carrier protein. So it's a carrier protein, okay? Now, the last one is bulk transport. Bulk transport is when the molecules that the cell want to transport is too big. How big is big? When the molecule want to transport is bigger than this protein, how can you use protein to carry something that's bigger than it? So you have to use bulk transport. That means the cell, for example, you want to take in the entire bacteria, right? Bacteria is made out of what? So many things, right? The peptidoglycan cell wall, the cell surface membrane, okay, and then all the components inside the cell, DNA, and so on. So therefore, cannot use carrier protein to transport. You have to use bulk transport. Means the cell will move towards the bacteria and then engulf it with the pseudopodia, right? So this is called bulk transport. So bulk transport is you take huge quantity of materials or huge quantity of material or large particle into the cell, all right? Either by phagocytosis or pinocytosis. So phagocytosis is a cell eating process, and this is pinocytosis is cell drinking process, right? Okay. So this one you learn bulk transport. This taking in is called endocytosis, right? So bulk transport is A, endocytosis. Then of course you've got B is exocytosis. Right, exocytosis, bulk transport material exit the cell, out of the cell after digestion, everything. Undigested material, unwanted material released by exocytosis. Right, so this bulk transport. Okay, so this is basically what we cover for this topic. Of course, there's one more very important one. Of course, under osmosis, we learn a lot, right? Under osmosis itself, extension from here, we actually learn a little bit more because it governs the movement of water into the especially plant and animal cell. So where can I do that? So here. Okay, osmosis. We learn about water potential, where I have to use symbol already, right? So osmosis is governed by this formula. Psi is equal to psi S plus psi P. Psi S plus psi P. Right? Where S is governed by the solute and P is governed by hydrostatic pressure, right? And S is always less than, psi S always less than 0, because the moment you add solute into the water, solute immediately attract water molecules. I should ask you a question then tell you, right? So therefore that will reduce, okay? Psi P is when it can be positive or negative, depending on whether it's pushing force or pulling force. Right, you take a syringe and you push the plunger, so it's pushing force, so it's positive. Then you take a syringe and pull the syringe, there's negative force, it's suction force. Right, positive and negative. So when these two combine, you add up, you give you a final psi. Psi will affect the movement of water, because water always moves from region higher to lower water potential. So then it comes to the definition of all these terms. You need to be able to define those terms. Right, what is simple diffusion, faceted diffusion, osmosis, right? Right, movement of particles from region higher concentration to lower concentration until the concentration becomes even throughout and equilibrium is reached. So that's diffusion. Then you replace it with water. Then you put it the partially permeable membranes, becomes osmosis. Movement of water molecules from region higher water potential to lower water potential until the water potential becomes even on both sides of the membrane. Right, or equilibrium is reached on both sides of membranes, osmosis. So definition also need to know. So we go into more detail on osmosis using this formula. And then we using this formula to explain what happens to the water, what happens to our cell, animal cell and plant cell, when placed in a water potential that is different or same, higher or lower than same as psi of the cell. So what will happen to the cell? So we learned that one. Hello, cell. Okay, so that was in detail, right? What else do we learn? So one more thing we learned is we learned a fixed law, right? Fixed law says that the rate of diffusion, which is such an important formula, is equals to what? By this time, I think all of you should be able to give me this formula. Okay, Amma, what is this formula? Please give me. What happened to my Ganyuhan? Plus 2 marks. Okay. Amma, give me the formula. Amar?
Yeah.
Okay, give me the formula for rate of diffusion. Faster.
Hold on.
Okay, so rate of diffusion is surface area times concentration gradient. Amma must remember this one. This is a very basic one. So divided by distance. So this is the formula, right? So therefore, you say that how come the rate of diffusion is higher or lower is to be governed by this one. And of course, I said one more thing that's not inside the formula is the temperature, because temperature governs kinetic energy of the molecule, so govern how fast the molecule can move, right? So surface area times concentration gradient divided by distance, okay? Right, so this is another thing that we learned from this chapter. So I think we have covered everything about this chapter already.